I'm Jim Al-Khalili, a professor of theoretical physics. But today, my brain isn't pondering equations, it's being scanned.
You have about 160,000 kilometers of wiring in about 1.5 kilos. So enough to wrap around the Earth 4 times or take you halfway to the moon. Goodness.
Like everybody else's, my brain contains about 100 billion neurons and over 100 trillion connections. That's more than the stars in the Milky Way.
This is what your head looks like inside.
So how did something so complex actually evolve? Spanning hundreds of millions of years, this story takes in the strangest of creatures. So there's a tiny brain.
Yes, exactly.
Now, thanks to the latest scientific breakthroughs, we can tell the brain's extraordinary story like never before.
And that's the first whisper of a revolution, a cognitive revolution.
It's an epic tale. With amazing twists and turns.
Look how quick.
Oh, my word.
As we build artificial intelligence vying to overtake our brains, I want to find out what makes the thing in our skulls so special. This is my brain, and this is its 600 million year story. To try to understand the beginning of my brain's story, I'm snorkeling off the west coast of Italy. This is home to some very strange-looking animals. But go back some 560 million years, and you'd encounter the common ancestor of us and many of these creatures. It had a rather special shape, probably a bit like this worm, with a head and tail and left-right symmetry. This animal was, like us, bilateral, but it had something else too. Many scientists now believe that this was the first animal to possess the basic building blocks of the brain. It was in an epoch known as the Ediacaran that these bilateria first emerged. To find out more about their early brains, I've come to meet some scientists who are working in these waters.
Okay, pull up now and we will collect everything.
They're pumping up the water from a depth of 10 meters up through this filter, which lets through all the tiny organisms that they don't want to catch, and it captures all the ones they want to study.
They're looking for zooplankton, animals like tiny worms that drift in water.
So this in here, it's hard to see, but this is zooplankton. We can analyze this under the microscope. Small algae will go through, and the zooplankton will stay in the filter.
Why is it studying this tiny worm helps us understand the ancient early evolution of the brain?
We have chosen to work on this worm because we were convinced that it has ancestral features, so that it would show us, for example, characteristics of the brain that are old, that have always been existing in animals.
Like the first bilateria, these zooplankton have a tiny cluster of nerve cells in their heads and a strand running down their bodies. These cells coordinate muscle movement and other organ behavior. And crucially, as well as coordinating, early brains evolved ways of sensing and responding to their environment.
And so it was. So the first thing we found about this worm is that it uses light-sensitive cells, photoreceptor cells. that it has in its brain, and these cells are the same to the ones that we have in our retina, the rods and cones. And this was a big surprise, a big finding, that these are the same photoreceptor cells.
So these are cells that are activated, that send signals to the brain when light impacts in them.
We have these in our eyes, so whenever we see light, these cells are activated and signal to the brain that there is light. And the very same cells exist in this worm.
Back on dry land, another member of the team shows me what these worms can tell us about early brains.
Aha! Here it is. So this is a young or juvenile.
Right. So how big is this? Is like maybe a millimeter or two?
This is 2 millimeters.
Okay.
And it has on the front a nice head. You can see the pharynx of the animal where they take up food. You can even see some movement inside these little dark dots, which are the eyes.
So it has 4 eyes?
It has 4 eyes, and you can see these dark speckles. These are the pigments of the eye.
Inside these simple eyes are molecules called opsins. When light hits opsins, they trigger an electrical signal which can make the worm move.
Can you show me how they react to light then?
Yes, so here you see the larvae swimming, and then we switch on a directional light stimulus on this side of the chamber. You see when the light comes on, and they start to move to the light.
Oh, I see. You're replacing sunlight with your artificial light and having the worm move towards it.
Yes, we provide this directional light cue here from the side.
The worm isn't seeing, thinking, or responding like we do, though. It has a simple nervous system that makes it move when light strikes its opsins.
What does that tell us about the early evolution of vision?
We think that the first eyes were just, in fact, very simple, a few cells able to tell the direction of light. And this kind of mechanism was probably there already in the common ancestor, last common ancestor of the bilaterian animals around 540 million years ago.
At about the same time, other senses developed too. For instance, cells that could respond to nutrients, the forerunners of smell and taste. But these first bilateral animals also needed a way of sending sensory signals around their bodies to control muscles and organs. For that, they used a special type of cell, the animal world's fiber optic cable, a nerve cell, or neuron. It's taken over 100 years for scientists to understand the neuron. Much of that story happened just down the coast here in Italy, at a true landmark in the history of science. This is the Stazione Zoologica in Naples. It was founded in the 1870s by zoologist Anton Dorn.
Anton Dorn was an early and passionate adopter of Charles Darwin's theory of evolution by natural selection. And that passion drove him to create a station for scientists to study marine life. For his location, he chose Naples, because, well, Naples.
Dorn built this impressive laboratory right on the beach, with easy access to the Mediterranean and facilities to study all kinds of sea creatures. To explore how neurons and nervous systems worked, he had his team make some exquisitely detailed and beautiful illustrations. Dr. Graziano Fiorito showed me some examples.
There was a strong tradition by Dorn of supporting the study of various organisms.
Wonderful. So these are illustrations made for this journal, which is Fauna and Flora. 1896.
They were interested not only in the shape of the animal, the organs, but the nervous system, how the nervous system is organized.
Then, in 1932, here at the Stazione, a British scientist called John Zachariah Young, or JZ, made a breakthrough in understanding how the neuron works. Scientists had previously suspected electricity was involved in neuron signals, but these cells were always too small and thin to really study. Then JZ found an exception. Squid possesses neurons so long and thick, they're actually visible to the naked eye.
This is a classic video where JZ was showing how to dissect a squid. He is now removing some part of organs, the gills, in order to reveal this network, you see?
Right. JZ realized that because these neurons were so large, he could isolate one with the correct dissection techniques.
So these are all nerves?
Nerves.
Each one of those lines?
Yeah, and finally, this is the giant single nerve.
Scientists used electrodes to measure what was happening inside the neuron. And this led to the discovery of precisely how electrical signals traveled along it.
Would you say this was the beginning of modern neuroscience?
Yes, JZ Young published this paper in 1939, and this was the emergence, let's say, of neuroscience.
We now understand that nerve cells, or neurons, are the building blocks of all brains. Neurons take signals from molecules like light-sensitive opsins and transmit them through the body as electrical pulses. But how exactly do they do that? Most neurons consist of three basic parts. Dendrites, these branching filaments that pick up the signals, a cell body that processes the signal, and a long strand called an axon that carries a signal to other cells. But what's really incredible is just how fast the signal can move. Let me show you the patella reflex, or the knee-jerk reaction. My director, Tim, has kindly volunteered here. So look what happens when I do this. Hitting the tendon triggers a nerve in the thigh, and a signal travels to the spine and back, and the thigh muscle jerks. Watch how fast it is. It takes about 18 milliseconds, which means the signal is traveling at about 200 kilometers an hour. Electricity doesn't flow along the whole length of an axon, as it might do in a copper wire. Instead, the signal starts when a tiny channel at the beginning of the axon opens, allowing electrically charged particles to flow through it. This then triggers other channels to open a little further along. It's rather like a domino effect. Each tile moves very little, but the ripple effect travels rapidly. Same in the axon. The charged particles themselves hardly move, but the signal races down. Ultimately, what we see is a pulse of electricity passing from one end to the other. And it all happens in the blink of an eye. But what makes the brain and nervous system so flexible is that neurons signal other neurons, creating a complex network. They communicate across a tiny gap called a synapse. Here, the electrical signal triggers the release of a chemical called a neurotransmitter, which crosses from one neuron to another. In this way, signals can cascade through your brain and body. And new technology means scientists can see and hear neurons at work. In Boston Harbor, on the eastern seaboard of the United States, a fresh haul of tasty crabs is just in. But before they reach the plate of a hungry diner, some of these crabs are being diverted for scientific research.
So what Kira's doing now is she's pinning out the sympathetic gastric nervous system.
Professor Eve Marder runs this lab at Brandeis University near Boston.
Once it's pinned down flat, she'll place recording electrodes on the nerves that will allow her to identify the neurons and record the motor patterns.
Eve's team are measuring electrical activity in the neurons.
These very fine white lines are the nerves themselves, and you can see how delicate they are.
And the technology here means those signals are converted into sound.
So I'm using the electrodes to record the activity of the nerves, and you can hear that the rhythm is going pretty fast right now. You're hearing that nice rhythm of derp, 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 derp. It's the circuit doing its thing.
This is a real-time representation of the crab's nervous system actually sending messages, much like the ones that control our bodily functions. But in an era of fear about climate change, with temperatures set to rise, the discovery also has a worrying aspect. Neurons are prone to fail as their surroundings warm up.
Every human neurological disorder is exacerbated by high temperature. The fact that poor people and ill people are less likely to be in environments where they can regulate their room temperatures and therefore their body temperatures means that we're potentially heading into a major health crisis.
This fragility means we should never take neurons for granted. After all, these cells carrying billions of pulses around our brains and bodies every second arguably make us who we are.
All the thoughts and all the dreams of the entire human race are just made up of these tiny electric signals racing along neurons. It's mind-boggling.
For neurons to become really effective and lead to brains like ours, they had to do more than simply carry signals. They had to take context into account, respond to stimuli in much more nuanced ways. To do that, evolution picked a group of ancient proteins and found a new use for them: neuropeptides. Let me give you a sense of the rather subtle ways these work.
You see, depending on circumstances, the same thing could be either good or to be avoided. Let me give you an example of how important this is even now.
Think about how you might respond to a delicious-looking cake.
If I haven't eaten for a while, then I really just want to tuck in. But if I've just had a big meal, then it looks a whole lot less appetizing. And if I've just been sick from food poisoning, then the same cake can look repulsive. Our brains have a way of adapting our behavior by reprogramming what the neurons tell us to do.
These subtle alterations of when and why neurons fire are controlled by neuropeptides. For instance, they change our behavior when we're with friends rather than strangers. Or how a mother acts when she's with her new baby.
All these new behaviors happen slowly over minutes or hours and are very different from the rapid response to, say, touching something hot.
Professor Corey Bargman at Rockefeller University in New York has made it her life's work to study how neuropeptides alter behavior and to understand how they evolved.
The neuropeptides are very old. This system is one of the first ways that the nervous system functioned, even before there was sort of a centralized brain, while it was still kind of fuzzy and distributed.
To understand neuropeptides, Corey studied a tiny worm known as C. elegans.
This is a single adult C. elegans, and here's a little adolescent next to her, moving around in real time. Because she's transparent, and under the right circumstances, you can see every neuron in her nervous system.
C. elegans has precisely 302 neurons. and scientists have now mapped exactly how each one connects to the others. Corey's research examines how neuropeptides influence this basic nervous system and enable the worms to have complex behaviors, like socializing or courting.
Some of the worms were very interested in associating with other worms. And we can actually see these behaviors here in this movie. And what you can see is that they're tending to hang out together in these little groups. And this is an aggregation behavior. Aggregation behavior is very common in animals. Animals often associate with other animals in their same species. That could be called herding behavior in sheep, or it could be called schooling behavior in fish. And we see exactly this even in these very simple worms.
Normally, when worms bump into things, they move away. But Corey found that sometimes something very different happened. A neuropeptide you might have heard of called oxytocin was released, which changed how the neurons work and how the worms behave.
Oxytocin is required for mating behaviors in C. elegans. I'm showing you here a mating pair of a male C. elegans and its female-like partner. And in the male, there's a single neuron. It's this bright spot here that looks almost like an eye. That's the neuron that is making oxytocin when the animal is mating. And so this male is resetting his behaviors so that in the presence of a mating partner, he'll respond appropriately for this for the next 5 minutes. And then once she disappears again, he'll go back to the other kinds of exploratory behaviors that he's doing.
But what really surprised Corey was how neuropeptides like oxytocin changed behavior in almost all living things.
We found that there was an oxytocin in ants, which of course are a social species, and it's involved in some of their social behaviors as well.
And in mammals like us, oxytocin has developed a crucial role.
Oxytocin is released when a female mammal gives birth. It helps to rewire the mother's brain to stimulate bonding between the mother and her offspring. It's being able to flip from one state into another. And we all know that experience in our own lives. We all know how emotional states are changing our behaviors on a moment-to-moment basis.
In our brains, neuropeptides control our moods, bonding, love, anger, sex. In fact, modern drugs like Ozempic, which is set to revolutionize obesity, is linked to the action of the neuropeptide GLP-1, which controls appetite. It's quite incredible that so much of our behavior is based on these proteins inside our brains that evolved hundreds of millions of years ago. The next huge leap in the brain's development happened about 530 million years ago, in what's known as the Cambrian explosion. Evolution went into overdrive. There was a huge burst of new, strange-looking species. Big creatures with shells, thorns, fierce claws and hooks. And one particular adaptation that would have far-reaching consequences for our brains.
I'm about to show you something really exciting. Hi, Philomena.
Hi.
Can I have a look at
Yes.
what's in that dish? Thank you. I'll put it on this dark background. What's inside here is really rare. These long creatures, that's called Amphioxus. It's regarded to be very similar to the earliest, the common ancestor, about 500 million years ago, that marked the transition to the first vertebrates. Maybe we can have a closer look under the microscope.
Here we have the adult Amphioxus. Here we can see the anterior part, the head. Here the posterior part, the tail.
And from head to tail, the nerve cord has been supplemented with a string of cartilage and bone, a primitive spine. This gave the ancient version of Amphioxus an advantage over earlier Bilateria. it would allow brains to get bigger.
So there's a tiny brain.
Yes, exactly. If we go more in, we can see here we have this enlargement. And here there is the brain of Amphioxus.
The rigidity of spinal cords and early skeletons allowed vertebrates and their brains to grow. Amphioxi have 20,000 neurons. You can get a sense of the advantages of having a smarter brain and internal spine and skeleton by watching this fish tank.
Right now, this looks like a perfectly peaceful habitat. Fish gently moving about, calmly, harmoniously. But that's because they're not thinking about their lunch. Let the feeding commence.
Vertebrates of all kinds proliferated in the Cambrian explosion. Thanks to their speed and maneuverability, the ones with spines and bigger brains were great at both hunting and escaping.
The main scan is basically ready.
At the MRI scanner at University College London, I'm being shown the debt my brain owes to my early vertebrate ancestors.
That's your spinal cord coming up into your brainstem. Brainstem is one of the sort of evolutionarily older parts of the brain that keeps your heart beating, your lungs pumping, move your eyes around. So lots of important things.
Another crucial component of vertebrate brains is the hippocampus, which we use for memory formation.
The hippocampus is a little C-like structure that shows up here. And I can see it because I'm used to looking at hippocampi, but it may be a little bit challenging. It's sitting just below your amygdala, which is part of the brain that's sort of an emotional center.
This new large vertebrate brain was matched by an increase in body complexity and size too. And this meant the brain and body needed more sophisticated internal communication.
Brains not only had to get better and better at sensing what was going on in the world outside, but also what was going on inside the body. And one very important way they do this is fascinating. They use a molecule that works a bit like a telegraph key sending Morse code. A telegraph key is pretty simple. A battery connects across a lever arm and a contact below. So when I press down on the arm, I complete the circuit, and a current flows through the wire, enabling me to send a message. In essence, a mechanical force, in this case, the pressure of my finger, is triggering an electrical signal.
For years, scientists suspected something similar was happening in our nervous system. Something was transforming mechanical force into some sort of signal to the brain. For instance, as a stomach fills, its walls stretch. And somehow the animal knew it didn't need to eat for a while. But how did this actually work? The answer was finally discovered in 2010 by Professor Ardem Pataputian at Scripps Research Institute in Southern California.
We're used to thinking about taste and smell as sensing chemicals, but this is actually sensing physics, right? It's mechanical forces, pressure. You have to turn this physical stimulus into an electrical or chemical signal that neurons would understand. And this transformation has been a big mystery.
The challenge was to find a molecule that, like a telegraph key, was triggered by mechanical pressure to send an electrical signal. So Ardem's team built an ingenious probe that gently applied pressure to cells just a few thousandths of a millimeter across.
We have a video here of the cell poking assay under high magnification. And so what we're doing here is using that to deliver submicrons, so very, very small fractions of a millimeter indentations to this cell that's sitting on this dish right here.
After nearly a year of poking and prodding, they finally found a single molecule on the cell's membrane which triggered a tiny electric current when pressed.
So when we poke very far into the cell, we see a very large current. And when we only poke the cell a little bit, we see a very small current, or none at all. And this tells us that the cells are really exquisite sensors of mechanical force.
They called this molecule piezo. This is a model of one of the molecules that Ardem and his team discovered, the biological world's telegraph key. It's embedded in the surface membrane of cells like the neuron. And it's tiny, just a few millionths of a millimeter across. But despite that, it's a really complicated molecule. It's a single protein made up of roughly 50,000 atoms arranged in this intricate shape, with 3 spiral arms and a cap in the middle. The shape of the protein is crucial. When the cell wall stretches, the whole structure flattens out, and the center opens up, creating a channel through which charged particles can flow into the cell. This is analogous to pressing the telegraph key, allowing an electric current to flow. We now know there are hundreds of thousands of piezo molecules throughout our bodies, signaling our brains. They can monitor our blood pressure, appetites, the need to wee, the timing of our heartbeats, and even regulate our breathing. And because hundreds of thousands of piezos are embedded in our skin too, they enable the most exquisite and underrated of all senses, the sense of touch. Think about it, our ability to touch is amazing. We can feel the tiniest breath on our fingertips, or the feel of a single grain of sugar, or the gentlest brush of a feather. Scientists say we can sense something on our fingertips that's just 1/500th of the width of a human hair. Having discovered piezo molecules, Ardem Pataputian's team then looked at where evolution had used them, and what they discovered was astonishing.
They seem to be pretty conserved across evolution. So we don't see them in bacteria, but in anything that has a nucleus, even unicellular organisms have them. We have seen them in flies, in worms. Plants, of course, have to sense mechanosensation as well. Roots, as they navigate through the soil and hit a harder surface, they have to decide to go through it, or if it's too hard, go around it.
The fact piezo molecules can be found across such a wide variety of life tells us they evolved a long time ago.
Our sense of pressure sensing that we mainly think of as sense of touch was present hundreds of millions of years ago in organisms that were just maybe swimming in the ocean and bumping into different things and needed to know what they were bumping against.
Up to this point, all life on Earth had lived and died in one place, in the oceans. The land was sterile and empty. But then things changed. Towards the end of the Cambrian explosion, about 500 million years ago, we think the first complex life began to creep out of the sea and onto the land.
Those first things that colonized the land were algae, mosses, basic bacteria. But then larger, more structured organisms began to emerge. Plants evolved root systems that went underground. Tall grasses emerged, and eventually trees.
Following them out of the water were early insects too, crawling and burrowing and wriggling things.
For sea creatures that lived close to the shoreline, the land now represented a new and potentially resource-rich environment. But of course, these animals would have to evolve considerably if they were going to survive above water. The process took millions of years.
You have to jump forward to 375 million years ago to see the first vertebrates starting to venture onto terra firma. This is a mud skipper. It's similar to the kinds of animals that were beginning to evolve at this time. Their eyes have moved upwards on their heads instead of on each side with most fish. Many scientists now believe that the way eyes worked out of the water led to one of the most significant turning points in the evolution of the brain. Professors Malcolm McIver and Dan Dombeck at Northwestern University in the States have been studying this critical moment in evolution.
With just the rising of the eyeball out of the water, you go from a medium which absorbs light so intensely that you only see a body length in front of you to essentially a perfectly transparent medium. That gives you a huge multiplication in your visual range.
Now, the key reason for that is to do with physics, or more specifically, the physics of light. You see, light travels far better through air than it does through water. That might sound rather obvious, but let me show you. Safety goggles on. What I have here is a tank full of seawater and a laser pointer that shines light through the water to be picked up by a detector on the other side, which I can read here.
When I turn on the laser, the light meter reading doubles from 25,000 to 50,000 lux.
That's how much light is getting through the seawater to the other side. Guys, give me a hand here. Okay, thanks, guys. Right, now we do this again.
We'll repeat the test, but with an empty tank. And the reading has jumped to over 100,000 lux. Air is at least 20 times more transparent than seawater. You can clearly see this when you swim in the sea. You just can't see very far underwater. That's because water is denser than air, and often murkier too because of mud or sea life. And this had huge implications.
As animals moved out of the sea and onto the land, the amount of raw visual information they could receive increased enormously, and their brains had to evolve to keep up.
How are you going to process all of that visual input to survive threats that are coming at you or to find food that might be trying to escape from you some distance away? So you need to have, you're going to have to up your computational brain in order to process all that information.
Malcolm and Dan believe that moving from sea to land led to a massive growth in size and power of the brain.
I want to demonstrate with these two skeletons just how big a change there was in relative brain size between ancestral fish and mammals. And so here is a fish that's in real life about this big, and its body mass a bit larger, but it's approximately similar to what a cat is. And yet, the total volume of this animal's brain is about 2 teaspoons, and it's a factor of 10 larger brain in mammals compared to fish.
The consequences of the move to land are stamped all over our brains.
Significant regions of the brain have evolved to make sense of the mass of visual data pouring in via optic nerves connected to our retinas. Regions like the primary and secondary visual cortex, the posterior parietal lobe, and the fusiform gyrus.
But though the move to land put down the foundations of these parts of my brain, it very nearly never got going, because around 250 million years ago, catastrophe struck. a massive series of volcanoes erupted in Siberia. The cataclysm exposed a huge geological fault stretching for hundreds of miles. And from this gash poured billions of tons of hot lava. Some suggest this flooded the plains below and came into contact with one of the largest exposed coal deposits in the world. Picture the scene 252 million years ago. Billions of tons of red-hot lava, billions of tons of thick, black, dirty coal deposits. The result was a toxic gas release on a catastrophic scale. Poisons like sulfur dioxide, nitrogen sulfide, and methanes filled the atmosphere. They then mixed with moisture in the air to create powerful acids, which then rained down, wiping out nearly all life from the face of the Earth. As well as this, the volcanoes belched carbon dioxide from deep inside the Earth. Trillions of tons, suffocating almost all plants and animals. Today, we consider the world to be on the brink of a carbon dioxide catastrophe. There are 420 parts per million of carbon dioxide in the atmosphere and rising. But 250 million years ago, the Siberian volcanoes released 5,000 parts per million, over 10 times more. There was total devastation. 95% of all species were wiped out. In the whole history of life on Earth, it's the closest we've come to total extinction. But of course, it wasn't the end. Some things did make it through. The survivors were smaller animals that could hide away, they could burrow underground, they could crawl into crevices or slither into the mud. They could survive on less oxygen. They could cope with the heat, the darkness, and the toxic chemical mix. The scene was now set for one of life's most incredible comebacks, and one of the most profound moments in the story of our brains. This is the Vale of Glamorgan in Wales. After the massive extinction event, this whole area was an archipelago of small, low-lying, linked islands, which makes it particularly interesting to paleontologist Dr. Ellis Newham. What was this place like back then?
It's 50 million years after the end Permian mass extinction, the Great Dying. So life was bouncing back by then.
We're now firmly in the era of the first dinosaurs, the gigantic creatures that were coming to dominate the world. But Ellis is more interested in a much, much tinier creature, one whose remains literally fell through the cracks.
Within these islands, there was cracks and fissures that naturally developed. And so any animal that died, their remains would have got washed in. And so this gives us an amazing snapshot onto what life would have been like on those islands 200 million years ago.
And what Ellis and his colleagues found deep in these rocks were new clues to how our brains were actually formed. Bones from a tiny rat-like creature called Morganucodon, or Morgie.
We found thousands of isolated teeth, jaws, and bones. And I've brought one here with me to show you.
I mean, it's tiny. This ain't no T-Rex. So what did this creature look like?
It was about the size of a shrew. It might have been furry. It probably had whiskers. And it was kind of scrabbling around the undergrowth in what is now South Wales.
Right.
So it was definitely insectivorous. It fed on beetles and kind of gnatwings and things like that were living with it at the time.
But what no one expected is what they saw when they started looking inside Morgie's skull.
Colleagues CT scanned on these skulls, and what they found was pretty amazing. So this is a cast, a 3D print of a model that they created of the brain of Morganucodon.
Wow. Very tiny.
Absolutely tiny. Maybe I'll get a scaled version for you.
Right.
So here we go.
On top of Morgie's brain, they noticed an intriguing new development.
We started noticing this thing. You can see this lumpy bit here.
And so the brains of other creatures that lived around that time didn't have this bump?
As far as we know. So Morgie shows a kind of a jump step in development of the brain size and also the brain complexity.
This bump is a new part of the brain called the neocortex, and it's what sets creatures like Morgie apart from other animals of the time.
It's a small bump with huge implications because it's the bump that makes our brains so unique. So where did it come from?
To find out more about the neocortex, I've come to meet Professor Steve Brussate, an expert in paleontology and evolution.
This is Morgie's brain. This is a digital model from a CAT scan. And just to orient ourselves, this bit here at the front, that's the olfactory bulb. That's the part that controls our sense of smell. Back here is the cerebrum, one of the main components of the brain. And up there on the top is that curious little bump.
And that's what I'm interested in. So where did that come from?
We have a few clues that we think helps tell that story. And the first one of those is if we look at the bones of Morgie.
Unlike any animals before this time, the microscope reveals Morgie's bones are honeycombed by tiny holes, evidence of a massive step change in blood flow.
Those are just digital renderings of the channels for blood vessels. And the key thing is that their bones, their skeletons, were becoming much more full of blood and blood vessels. And that means that they could have higher metabolic rates.
So their bodies were just working harder?
Their bodies were working harder, they could grow faster, their metabolism was increasing. That's part of the story. But there's a second clue to this as well, which we'll have to take a look over here.
Okay.
And this is a little fossil discovered about a decade ago by teams from here at the National Museum of Scotland, out on the Isle of Skye. And if we put it under the scope, we can see what it is. The bit sticking up from the top, that tells us what this fossil is. And those things are teeth. So this is a jawbone. But when you blow it up in size, you 3D print it, you can see all of these teeth here, and it's in these teeth that tell us the importance of these fossils. And they tell us a few things. These teeth, they're really sharp. There's lots of little points. And when the teeth came together, all those points would interlock, and they would bite really strongly, be able to chew the food. But the other thing is, the babies did something different. We can tell that these animals had one set of baby teeth, one set of adult teeth. And that means that the babies were drinking milk.
Okay. And what does that tell us?
So these, of course, are mammals, just like us. These are early ancestors of ours. What we see with these fossils at this time, these mammals were entering an age of gluttony.
So these mammals ate more, their offspring drank milk, and they had more blood flow in their bones. This wasn't just the start of mammals, it was a new way of being.
That tells us the big reveal here, and that is that we think that these mammals were becoming warm-blooded. And that means you have a constant body temperature, a high body temperature, and your body temperature is high and constant regardless of the season, regardless of the time of day, regardless of where you live. And that's a very special superpower. That's what we have.
Warm-bloodedness enabled animals to exploit new niches, hunting at night and in the cold.
So it takes resources to be warm-blooded. If you think about the cost of heating our homes, it's expensive.
A mammal needs to eat about 8 times, give or take, as much food as a cold-blooded animal of a similar size.
So bringing this back to the beginning, how does all these changes affect the brain of Morganucodon?
That's where that bump on the brain comes in. And that's the neocortex. It's clearly getting larger. That's the expansion of the brain that's happening at the exact same time in evolution as these changes to the teeth, changes to the diet, changes to the metabolism.
So you have the evidence, the bones, the teeth, suggesting they are eating more, they're metabolizing more, they have to find food more often, and to do that, they need bigger brains.
That's it. That's the story, and it's a huge story, and it's one of the biggest changes that we see in the evolution of animals.
So what did these new parts of the brain allow animals to do? It seems to have led to the development of a remarkable new ability, a true superpower. That superpower is what we might loosely call imagination, our remarkable ability to remember scenes from our past or to conjure up ones in our future. Cast your mind back to when you were a young child. Try to remember what it was like. Now conjure up your next Christmas or family gathering. Can you see in your mind's eye what it will be like? What's amazing is how effective this internal visualization can be, our ability to see the past or imagine the future. We can link it to smell and even to our emotions. To investigate the mammalian origins of imagination, Malcolm McIver and Dan Dombeck built this, an open maze with food at several reward ports. And because genuine early mammals no longer exist, his team used mice to see how they visualize the world around them. And to help motivate the mouse, an unusual predator.
We came up with a large arena where we could essentially run a predator-prey scenario, but with the predator being an autonomous robot that we have designed and developed. The robot doesn't physically attack the rodent in the space, it simply puffs an intense blast of air. And rodents really dislike that, so they avoid the robot.
The team then attached probes into the mouse's hippocampus, which processes location information from the mammalian neocortex.
And we asked what exactly is going through the mind of the mouse. Is it imagining a future position of the robot? Is it imagining a future position of itself?
They put the mice through the maze thousands of times, an environment specially designed to make them think ahead. They then took data from the probes to get a sense of what was happening inside their brains.
So here's the experiment that you saw earlier. We've also included neural data.
The blue dot represents where the mouse actually is, and the red dot is where the robot is. But incredibly, by reading the signals from the mouse's neurons, the team could tell where the mouse was planning to go. That's the pixelated blue blob. Malcolm believes this reveals important clues about the mouse's imagination.
You can see that essentially we have the mouse thinking about where it's getting to in the next little bit. It's actually here, and this is where it's going to be soon, and it seems to be thinking about where it might be in the future. So right now, the mouse imagination of its location and its actual location are very close.
But then something remarkable happens. The pixelated blue blob moves away from the blue dot. This suggests that the mouse is thinking of where to go next to find food.
Right here, it skips over to here, and it starts to coalesce around this next reward port, which you'll see in a second is where it's going to go to next. So it appears to be thinking about, this is a reward port, and it appears to be thinking about the next reward port it's going to be visiting. And that is really interesting, because watch what happens after the mouse leaves this position, after it's checked that reward port. It actually goes over to exactly the location it seems to have been thinking about. So it's thinking of the future, it's imagining the future, which is exactly what we'd hope to see with these more sophisticated behaviors that the arena is exhibiting.
This is a bold claim, that leaving water for land, where long-range vision makes planning a matter of life or death, may have led to the ability to imagine.
If you're going to do the more complicated thing, which is essentially representing the future and future opportunities and threats, that requires a lot of neural power. It's a massively intensive task. So this is one of the demands on the nervous system that presumably feeds into this huge 10x increase in brain size in the mammals and in the birds relative to ancestral aquatic organisms.
Being able to visualize the future, to plan and solve problems, powers enabled by the burgeoning neocortex, would eventually come to give mammals a huge evolutionary advantage.
They survive, probably in large part because of their large brains, their high intelligence, their ability to solve problems. Within a few 100,000 years, we start seeing mammals the size of pigs. Within about 1 million years, we start to see mammals the size of cows. So as these mammals themselves are getting larger, the brains are getting larger, but you can see it better on CAT scans. These are the brains, you can kind of see them getting bigger, and that gray bit at the top, that's the same thing as the bump in Morganeucodon. That's the neocortex, but you can see it's getting bigger and bigger and bigger. And as mammals continue to adapt and evolve into dogs and cats and bats, whales, elephants, kangaroos, all the mammals that we know, monkeys, apes, and eventually humans, that neocortex, it came to envelop the surface of the brain.
And so most of what we regard as the brains of humans actually emerge from that bump in Morgie.
Exactly. And that's the first whisper of a revolution, a cognitive revolution. And we see in ourselves that same structure, but at just much grander scale.
So how important would you say was the emergence of warm-blooded animals in the evolution of the brain?
To me, that was the single biggest Big Bang instance in the history of brain evolution. So much about the brains that we have today was shaped by our metabolism being evolved by our fossil ancestors.
If Malcolm McIver, Dan Dombeck, and Steve Brusate are right, then the movement to land and the development of warm-bloodedness are responsible for the single largest neural growth spurt in 600 million years.
It gave us the greatest gift of all, the brain.
Next time, foraging, tool building, child rearing, and relationships. How our social lives have shaped the human brain. Curious about how MRI technology can reveal the secrets of your brain? To watch the Open University's new animation exploring how an MRI works and what it reveals, scan the QR code or go to bbc.co.uk/secrets of the brain and follow the links to the Open University.